I will let the floor now to our two chairs from this afternoon, Dr. Song Wang and Professor Sara Cherry.
Hi, everyone. Welcome to this afternoon session. It's a great pleasure for me to introduce Professor Sara Cherry. Professor Sara Cherry is a professor of microbiology in the Department of Microbiology at the Perelman School of Medicine of the University of Pennsylvania. She is also the scientific director for the core facility of high throughput screening, the director of program for chemogenomic discovery at the Penn Center for Precision Medicine. Dr. Cherry has published over 75 peer-reviewed articles in journals such as Cell, Nature, Immunity, and Molecular Cell. She also serves on multi committees for multi organizations such as NIH. So now I will give the floor to Professor Sara Cherry. She will present her recent work on SARS-CoV-2. Thank you. Welcome.
Thank you very much for that kind invitation and kind introduction. It's very nice of you. So what I'd like to talk to you today about is the work that we've done over the last year trying to apply high throughput screening approaches to identify antivirals active against SARS-CoV-2 and how we got to using diverse respiratory models, really because it's required in order to really understand how this virus infects humans. So obviously I don't need to belabor the point here, but over the last year and a half, this virus has spread across the globe, causing a pandemic with hundreds of millions of cases and many, many deaths. And we're really fortunate that vaccine platforms came online very quickly, and this is already outdated. There's been more than 2 million vaccines given globally. And so that's amazing news for all of us. However, as we know, there are major variants spreading across the globe, some of which can be protected by the vaccines, others are uncertain. And so we really need to continue to fight against this virus. And in addition, the SARS-CoV-2 is one of a family of coronaviruses that continue to emerge and reemerge across the globe. And so we're really interested in understanding how these viruses can infect humans and whether or not we can develop therapeutics that might be more broadly active against groups of coronaviruses to help us in the future. So I just want to tell you about the types of antivirals that we're trying to identify. So there's really sort of two classes of antivirals. There are the direct acting antivirals that actually target a viral protein or a viral part. And those are basically what almost all of our current antivirals target. The viruses carry enzymes that are required for replication, like RNA-dependent RNA polymerases. And so if you can target those, you can block viral infection. In addition, viruses, in order to grow within cells, have to hijack and subvert cellular pathways. And so we and others are really interested in also identifying host-targeted antivirals. That is to block some part of the life cycle that's required for infection and thereby blocking viral infection. So a good example of that is entry factors. There's been a lot of efforts to try to block viral entry. And then there's another class of host-directed antivirals, which are called immunomodulators. So that is if we could boost the immune response against the virus, for example, in the early recognition of viral infection, then maybe we could block viral infection as well. And so I'm going to tell you a little bit about an immune modulator we identified and then where we are with the host-directed antivirals. Okay. So actually early on, and this is really why we became very interested in studying respiratory cells, what was clear is that there was a lot of studies in African green monkey cells or non-lung cells such as hepatocytes, which had suggested that the virus goes through a pathway that's dependent on a low pH compartment for the activity of cathepsins, which are acid-dependent proteases. And so, for example, if we take vero cells or hepatocyte cells and we treat them with the cathepsin inhibitor or hydroxychloroquine, we can block infection. So in all my graphs, the blue line will be infection and the green line will be toxicity. So the cathepsin inhibitor can block infection in the absence of toxicity. However, subsequent to these very early studies, it became clear there was a second way that the virus could enter cells, and that is by taking advantage, instead of this acid-dependent protease inside the cell, a cell surface protease that could cleave and trigger the virus from the outside. And so when we looked in lung cells, what we found is that, in fact, the cathepsin inhibitors and hydroxychloroquine had no activity, but instead these TMPRSS2 inhibitors could block infection and, in fact, lead to more replication of the cells. So the respiratory cell that we've been using for these screens are called Calu-3s. And so that led us to think that there were really two independent pathways for entry. And given that the fact that the virus causes respiratory infection, this led us to suggest that this pathway was important. And in fact, one could be misled by screening in these other cell types and looking for inhibitors in these other cell types. And indeed, if we look at our RT-qPCR assay looking at viral RNA replication, we can see if we treat with Camostat, this TMPRSS2 inhibitor, we can prevent infection in these Calu-3 cells, in IPS-derived AT2 cells from the lung. We could block infection, as well as in air-liquid interface cultures, bronchial air-liquid interface cultures from MatTech. So basically what this told us is that if we wanted to find antivirals that were active against SARS that might work in vivo, we had to focus on respiratory cells and not other cell types to do our screening. So this is the screening platform that we have set up at Penn. So we take these human Calu-3 cells and we plate them in 384 well plates using liquid handling robots. Then we use additional robots to add drugs into the wells. We do it with pin tools at 50 nanoliters per well. Then we bring these plates into the BSL-3 where we can infect them with SARS-CoV-2. And then we allow the virus to replicate and we look for wells where there's an effect on infection. So we read out the assay by microscopy where we stain the wells for a viral antigen in green and we stain the wells with DAPI in blue. So basically this allows us to see the number of cells in a well so we can get rid of any toxicity, any drugs that have caused toxicity, and look for antivirals that go from this level of infection to no infection, okay? So this is the assay that we've been using with live BSL, the BSL-3 version of the virus. So then we've been screening a number of different drug libraries. So of course, we bought all the antivirals that are published in the literature, including remdesivir and this EIDD-2801 or molnupiravir. We have our own in-house repurposing library of about 3,500 drugs. We partnered for Medicines for Malaria Venture. They gave us about 500, maybe 1,000 drugs. We partnered with the National Center for Advancing Translational Science, which is the NIH screening operation. They gave us another 2,500 drugs. And then we partnered with the Gates Foundation, who helped us to get the drug library from a company called Caliber, called Reframe, another 12,000. And then we created another library of microbial antigens, another 800. So using the screening platform, then we developed a pipeline for how we were going to move forward on small molecules. So we screened about these 20,000 drugs, and then we used various criteria for characterizing the candidates. Then we take the candidates and we do these dose response validation where we repurchase powders and we look to see if there's antiviral activity in the absence of cytotoxicity. And that led us to about 150 drugs. So from about 20,000 to about 150 that have activity outside of the toxic range in these Calu-3 cells. So now the question is, what do you do with these 150? And so what we've been doing is moving these into primary respiratory models and doing mechanism of action studies to figure out how they might be working. So the primary human respiratory cells that we've been using are these air-liquid interface cultures. And so, as you know, the virus can affect the nasal epithelium, the bronchi, as well as can go deeper into the lung. And of course, each of these cell types are similar, but not the same. And so we've been working with Felix to look at the nasal epithelium, where in addition to getting these cells in collaboration with them, they've also been running toxicity assays. So that way we're making sure we're using the drugs at the appropriate concentrations. We've also been working with a company, MatTech, to get the bronchial epithelial cells, again, so we can look to see if the drugs are active in these more relevant models. And on the side, we've also been doing some work with these human iPS-derived AT2 cells that we can obtain from a core at the University of Pennsylvania. And so we're sort of moving our drugs from the Calu-3 into these different models in order to really identify drugs that are active that in a way that's more reflective of the true infection in the human. Okay, so then once we find drugs that are active in respiratory cells and we understand a little bit about their mechanism of action, we've been moving drugs into preclinical models, either with hamsters or mice in collaboration with a number of our colleagues. With the idea that once we can demonstrate activity in these models, we can move into clinical trials. So let's start by telling you something from this screen here. So many of you are aware that the virus is quite good at hiding from the early immune system. And the idea is that this ineffective early response allows the virus to establish infection within the respiratory tract. And so if we could boost that early immunity, we might be able to block infection. So the idea here is the early response is low, but if we could boost that, we might be able to bring down viral load and thus, of course, mitigate pathogenesis. So our first step to understand that was to treat cells with interferons. So interferons are cytokines that are produced early upon recognition of infection and can block many different viral infections. And so what we found is that if we treat these Calu-3 cells with either type 1 interferon beta or type 3 interferon lambda, we can reduce infection as measured by viral RNA loads. However, you can also see that type 1 interferon is much more active than type 3, okay? But this suggested that indeed, if we could boost early responses, so we give this right before infection, that indeed we might be able to mitigate viral replication. However, interferons are proteins and they're difficult to deliver and certainly difficult to scale globally, so they may not make the best drugs. We thought we might be able to screen for small molecules that would activate type 1 interferons, but that would be more drug-like. And so what we did is we took this library of 75 what are called PAMPs, or pathogen-associated molecular patterns, or microbial antigens that can induce type 1 interferons and other related innate immune pathways. And so we screened these PAMPs in Calu-3 cells and identified a number of candidates. And the ones circled here are two related cyclic dinucleotides, which are known to activate STING. So we validated that using these endogenous STING agonists, CGAMP, we could block infection in the absence of toxicity, although the EC50 is quite high. It's like at 7 micromolar. Indeed, we can also show by this qPCR assay that we could significantly reduce infection more than 100-fold. But again, these don't make great drugs because their IC50s or their activity is quite high level. But a number of pharma companies have developed small molecule STING agonists that are much more potent and have better drug-like properties. So one of them is the STING agonist called DIBZI that was developed by GSK. And you can see here that now we have amazing activity in the absence of toxicity in these Calu cells, again, with very substantial reduction in viral replication. So we were interested in moving this into a more relevant model. And so in this case, these are the air-liquid interface cultures from MatTech, where we could show that we can reproducibly infect them and completely block infection by treating them with this small molecule agonist. Indeed, you can look by qPCR. We can see a huge reduction in infection when we treat the cells with the DIBZI, which is similar to the level of reduction that we see with interferon beta. Hopefully you can notice here that interferon lambda is actually more active in the primary cells than it was in those Calu-3 cells. Again, speaking to the importance of looking in these models at how things are behaving. Another paper also came out using the same inhibitor in Epithelix's bronchial air-liquid interface cells, and they found similar results. Interestingly, and again, why I stress these air-liquid interface cultures is actually if we took these human iPS-derived AT2 cells that in many ways reflect the endogenous epithelial cells, actually while remdesivir is active, this DIBZI has no activity, and that's because these cells have lost STING activity because they're in culture for a long time, whereas these more primary cells have not lost any of these important signaling pathways. Okay, so then we moved into an animal model here. And so this is a mouse model where we basically gave a single intranasal dose of this drug. And we could see potent activation within 6 hours of treatment of gene signature consistent with type 1 interferon. And if we compare that, the lung data from the mouse to the Calu-3s, we see very, very significant overlap, suggesting that these Calu-3 cells are modeling much of the infection in vivo. So then we challenged animals with SARS-CoV-2. So we took the Washington 1 strain, and this is in collaboration with my colleague, Mike Diamond. He did these at WashU, and basically give an intranasal dose of the SARS coronavirus in the presence or absence of DIBZI. And this is a ACE2 transgenic model. So what you can see here is that while we have an early weight loss, probably due to the 2 doses of the drug, we see that the animals are completely protected and actually they survive the infection, whereas these animals all die. And you can see, this is at day 2 and at day 4, that we have a substantial protection from infection in the lungs, like a 4-log drop in infection from treatments. Given all the emerging coronavirus variants, we were also interested in this would be active against another variant, which is the South African variant, 1351. So in this case, this virus can infect wild type animals intranasally. And so we showed that we could protect from infection of this strain in Calu-3 cells, and so moved into the animal model. And in this case, what we could show is that a treatment at either -6 hours or therapeutically at +12 hours, we could mitigate the weight loss by the virus, and we could protect from infection as measured by viral titers, suggesting that this strategy of boosting early innate immune responses may in fact be a tenable strategy moving forward. And we published this paper back to back with Kate Fitzgerald's lab, who's also made very similar findings. So of course, we're continuing to work on that, but I just want to get back to some of this other 150 or so drugs that we identified in Calu-3 cells as antiviral, and just give you a little bit of where we're going with this and what we've found. So here's a pie chart of the distribution of the classes of drugs that we identified, put into categories that are somewhat intuitive. And so one of the things that we found are a number of nucleosides. So we'll get more into this in a minute, but nucleosides are obviously the building block for the viral RNA. And of course, the only approved small molecule is remdesivir, which is a nucleoside analog that is incorporated into the viral RNA and causes chain termination. Okay, so we were interested in this class because obviously remdesivir is within this class, as well as EIDD 2801 or molnupiravir, which is being developed by Ridgeback and Merck right now as another nucleoside analog that causes RNA-dependent RNA polymerase issues. So they fall into a number of different categories. Most of them are purines, which we think is interesting, but we're not really sure what that means. And so we looked more carefully at all of these antivirals and whether or not they were active in multiple cell types. So I told you at the very beginning that we know that the entry pathways are very different in respiratory cells and non-respiratory cells. And what it looks like to us is that's not just true for that class of small molecules, but other classes as well, as exemplified by this data. So Calu-3s are on the top, and you can see that remdesivir, molnupiravir, and a number of other purines that are used largely in cancer and immunosuppression are active, and these are the ones we validated in Calu-3 cells, so not surprising. But then we also tested a cell line called A549s, which are another human respiratory cell line that don't naturally express the receptor, so you have to overexpress the receptor for the virus in order to get infection. What you can see here is that remdesivir is still very active, molnupiravir a little bit less, but active, and then it starts to look different. So here there's no activity because there's toxicity, and then here these two are also active. And then if you go to intestinal epithelial cells or hepatocytes, it gets more and more divergent, where again, remdesivir seems to be active in all of them. to a lesser extent molnupiravir, this other orally active small molecule. And then it's completely different in these different cell types, again, whether or not they're active. And so this really, again, speaks to the importance of doing the experiments in cells or models that you really think reflects in vivo. Okay. And then we looked by qPCR, right? This other assay, this viral RNA replication assay, and we compared these Calu-3 cells to, again, these iPS-derived AT2 cells, where you can see, again, there's some drugs are active in both cell types, and then others, for example, mercaptopurine has no activity in AT2 cells, right? And so again, we're trying to use these different models to sort of narrow down which drugs we think we want to move forward on. And just to show you, in the epithelics derived nasal air liquid interface cultures, again, we can see a big reduction with remdesivir, right? A more modest but reproducible reduction with EIDD 2801. And then these other two nucleosides that were active in Calus show no activity in these cells. So of course, we wouldn't want to move forward with these. And again, we're using this as a way to stratify our drugs and to move them forward.
So,
however, yeah.
You still have 5 minutes. That would be okay?
I have what?
5 minutes.
Oh, that's fine.
Okay.
And so this led us to, first of all, you know, obviously these two are being moved forward clinically, and so that makes sense, but also really shows us the importance of doing these studies in appropriate cell types. So this led us to ask, well, these two nucleoside analogs are actually two different nucleosides. So remdesivir is an analog of adenosine, or A, molnupiravir is an analog of cytosine. And so this led us to ask, if we combine them, would they be synergistic? And so here is our 2x2 analysis looking at the percent infection as we increase the concentrations of molnupiravir or increase the concentrations of remdesivir. And then we can use something called Bliss analysis to ask if there's synergy. So the null hypothesis is that they're additive. And indeed, you can see it's all green, which is in the middle here, which is additive. So mixing these two drugs, obviously, it's good that they're additive, but it's disappointing that they were not synergistic. But we're trying to identify other synergies between different drugs in order to, again, elevate them to move forward with into animal models and potentially into the clinic. So basically what we're trying to do is sort of integrate our small molecule screening into the biology of the viruses. From about 20,000 drugs, we identified about 150 antivirals. We're trying to figure out how in the life cycle they work, how they fit into metabolism, either of the virus or the cells, as well as how they fit into these innate signaling networks by using other omic approaches to really try to stratify them. And so we're working on a number of the other classes of drugs, as well as doing 2x2 synergy testing in order to elevate them and move them forward into these air liquid interface models, and then of course into animal models. Okay. With that, I'd just like to thank the members of my lab who contributed to all of this over the last year and a half when we started working on SARS-CoV-2, High Throughput Screening Core, as well as all of our collaborators that have provided us small molecules, funding, and technical advice, as well as the various cell models. So we've been getting cells from MatTech. We've been working closely with Samuel with epithelics, as well as some other groups within Penn, trying to really see how we can leverage these fantastic model systems to understand the biology. And all of our animal models that I showed you today were with Mike Diamond, but we're collaborating with other folks to test other classes of antivirals in animal systems. And with that, I'll take any questions. Thank you.
Well, great talk, Sarah. Thank you very much. So we have some questions from the audience. And I think the first question is directed to STING pathway. Honestly, we have two questions on this. And the first is, does SARS-CoV-2 inhibit STING pathway?
Yeah, we tested that. We don't see any evidence that SARS-CoV-2 itself can antagonize the STING pathway during infection, at least in the Calu-3 model where we've tested it.
And the second question, which is related, is what is a therapeutic window for STING agonists?
Right. So that's a really important question. So we delivered it intranasally, okay? So there's been no work in humans with intranasal delivery. So in fact, these drugs were developed for cancer and in large part actually given intratumorally. So, so that's one issue. The other is just like interferon, right? So this works through interferon. It can activate interferon pathways, which of course can ultimately have side effects. But in our cases, actually, we've shown that a very low dose given only once is completely protective. And so we think that that would be tolerated well in humans. But the issue really is, how do you know when to give it to people? Because what you wouldn't want to give an activator of early interferon to patients that have progressed to, for example, hospitalization, where they have a hyperactive immune system. So what you would really want to do is give this to people the second that they had symptoms, right? Very early, or even sort of prophylactically, or for, if people were exposed and were uncertain if they got infected. But you would only want to give it very early in infection, not late in infection.
Okay, so there is another question, thank you. So may you shortly summarize how your model cell lines, primary cells, IP cells varied in terms of infectability?
Yes, they do. So the CALU cells, the way we maintain them, we can hit like 75, 80% of them routinely. That's not true. It depends on how you maintain them, whether or not that's one issue with this question, is it really depends on how you maintain the cells and how you work with them. For the air-liquid interface cultures, we find that the older they are, the more permissive they are, the more they're beating, the more differentiated they are, both for the nasal ones and for the bronchial ones. If you use them too early, they have very low permissivity. So it seems that really a mature epithelium is what we need for our experiments. The AT2 cells, what we do is we have them grown in spheres, and then to do our experiments, right, we want the apical side facing out, which they're not when they're growing as organoids. We actually replate them after we disrupt them on a 2D model a few days after they've been disrupted. And we can't hit more than 20% of them. So the infection is not that high, probably because of what we have to do in order to access the apical compartment.
And do you see differences in the new virus variants? And if they are similarly infectables in terms of different models, and if they are also, if some models are more responsive to these variants?
Right, so we're just doing that now. We now have the original strain, the Washington one. We have the South African variant, as well as the UK variant, and we're doing all those comparisons now. But there's lots of experiments out there that suggest that they're very similar in how they move through cell culture and how they infect animals. There may be subtle differences, but I think probably they're largely very similar. But we are doing those experiments ourselves now, but we haven't compared them all side by side. So far, they're all sensitive to the sting agonist, but given the mechanism of action of that, perhaps that's not surprising.
Great. I think the last questions for sake of time. So did you test around the 150 molecules that you really clearly showed, which have been active? So has they been tested in Syrian hamster or other, let's say, models to show their promising effect in vivo? And did they decrease viral infections or viral particle load?
150 is too many, that's the problem. So that's why we're going from the CALU cells to the air-liquid interface cultures to basically narrow it down, and then things that are active in the air-liquid interface cultures move into mouse models and hamster models. So we've tested a handful of additional drugs in either hamsters or mice, and some have been active and some have not. And the ones that are not active, we're now going back to figure out if it's because of PK issues or because there's very little known about how any of these molecules work in hamsters, because hamsters are not classically used in general. So, but some of the drugs we found in the CALU cells that were active in air-liquid interface cultures are also active in the animal models. And those are the ones we're most excited about. But unfortunately, I can't get anyone to test 150 compounds in hamsters.
Thank you very much. So I think we should stop it for let's say this time. And there are still questions in the Q&A section. If you want to answer them directly, Sarah, please do so. Okay.